what we're actually going to talk about today. Quantitative genetics. So thus far this semester, we've really focused on qualitative traits. So qualitative traits are traits that have a discrete phenotype, right? So you're either tall or dwarf, right? You're purple or white. You are wrinkled or round, right? You're one or the other. There's no kind of range in phenotypes. It's just kind of a yes or no situation. So if you look here at the top, we have the dwarf and the tall, and then two pure breeding lines. We cross them together, and then all of the offspring are showing the dominant phenotype, in this case tall, and then we cross those heterozygous F1s, and we get the 3-to-1 ratio of dwarf to tall, right? So a discontinuous characteristic where you're just one or the other. However, that's not the case for a lot of things. A lot of phenotypes are not put into two discrete categories. We have a variation throughout a lot of categories, right? So in corn, you can have like a short-eared corn and a long-eared corn, but when you cross them together, you don't get all long-eared corn or all short-eared corn. You get a range of corn. And then when you cross those F1s together, you get an even larger range of corn. So these are called quantitative traits. So they are continuous. We have a range of phenotypes, a range of phenotypic characteristics that we see in our population as opposed to just a single one. So can you think of any human traits that are likely under quantitative control? So height, that many people thought height, skin color, hair color, eye color, cholesterol levels. I'm not sure that hair length in humans, but maybe in other organisms, there's a hair length that could be quantitative, potentially. Weight, toe length. So those are all really good examples. I'm not sure if diabetes, I'm not sure that that would be a quantitative trait, although risk of diabetes might be, because diabetes would be the discrete phenotype that yes or no you have it, but the risk associated with it, so some people may be born with higher risk factors and some people may be born with lower risk factors, so that might be a quantitative trait. Okay. So that is what we're going to focus on today. These quantitative traits, how do we figure out what they are? How do we figure out what genes are involved in regulating these quantitative traits, right? How do we figure out what traits are quantitative under genetic control and what traits are under environmental control, right? So here is a picture of a university where they lined all of the men up by height, right here in 1920, and then they lined all of the men up by height in 1997, and by 1997 they had begun admitting women, so now we also have women in our distribution. So what do you notice about the distribution of men versus women in the picture from 1997? So there's a lot of women that are 5'6", but then probably the average of the men's height is what? A little taller, right, 5'10". So the average height of women is a little bit lower than the average height of men. What do you notice about the average height of men in 1920 versus in 1997? They've gotten taller over time, it looks like. So over time, the phenotype has changed. We've gotten a little bit taller, right? So we see in 1920, the average height looks like it was closer to 5'7", and then over here, it looks like we're closer to 5'10" or so. So when we're thinking about these quantitative traits, what we're really going to be looking at is the variation we see in the population. So this trait, height, there's a lot of variation in this population. So what is responsible for that variation? Is the variation in the population due to genetic differences in all of these individuals, or is it due to environmental differences in how these individuals were raised and what they were exposed to and what they ate and that sort of thing? So that's what we're going to be thinking about, is the variation in the population due to genetic or environmental causes. So we're really focusing on variation. Right? Okay, so looking at variation, we want to start by talking about this. This is a tobacco flower. I don't know if you've ever seen one before, but they're kind of pretty. And you see that they have this tube here of the flower. And that tube length is actually variable. And so one study was looking at this variability in this tube length of this flower. So we had one strain where the average flower length was about 40 millimeters. And we had another strain where the average flower length was about 93 millimeters. Now these two strains are each pure breeding. So what does that tell us about the genetics of those strains? What does pure breeding mean? Homozygous. So every flower in this strain A has the same genetics as every other flower, right? But we still see some variability in our flower length. So what does that variability due to? It's not to genetic differences, it has to be due to the environment, right? So it's due to the environment. So in this example with strain A and strain B, all of the variation is due to the environment. And the environment may lead to epigenetic changes in the DNA, right? We've talked about that a little bit. But we're just going to stick with kind of thinking of it as changes due to the environment. So if we take strain A and cross it to strain B, we get our F1. What do we know about the genotype of the F1? It's heterozygous, right? So all of the flowers in the F1 generation, again, are going to have the same genotype. They're all heterozygous. But again, we're seeing some variation in the flower length. So all this variation is due to the environment. But what if we took those F1s and crossed them together? Would our F2s all have the same genotype? No, we're going to have a range of genotypes, right, in the F2. So now our variation is due to both the environment and the genotypic differences, right? You following me so far? So looking at these sorts of experiments, he determined that these genes that have this variation that we see that it increases when we have this genetic diversity probably is due to the idea that the phenotype is due to the additive effects of a number of different genes. Okay? So if you look up here at the very top example, right, this would be like we talked about what the very first or second day of class with like the purple and the white and the flowers, right? You could be big A, oops, you could be big A, you could be big A, little a, you could be little a, little a. You have three different phenotypes. Then if you look at the second example, right, if we have 2 loci controlling a particular trait, now we can have 5 different genotypes. But if we have 5 loci controlling a trait, now we have even more possible genotypes. And if we have lots of loci controlling a trait, we have a lot of different possible genotypes, which leads to a lot of different phenotypic classes. So the effect of each gene itself is small, but when you add them together, they have a large effect. And then we say the environment also plays a role. It'll smooth out your curve, right, and it plays a role in the expression of the trait. So that makes sense. The more genes involved in a trait, the more variety of genotypes we can have. Therefore, the more variety of phenotypes we have. So let's just walk through an example to kind of put some numbers with our phenotypes and genotypes. Okay, so suppose we have 2 genes involved in controlling plant height. Obviously, there's probably a lot more, but we're just going to stick with 2. The genotype of little b, little b is 10 inches tall. Each dominant allele adds 5 inches to the height. So how tall is a plant that's big A, big A, big B, big B? 30 inches, right? We start with the 10, and we add 5, 10, 15, 20. 10 plus 20 is 30. Okay, if we take a homozygous dominant cross to a homozygous recessive, how tall is our heterozygous F1? That's going to be big A, big B, little b. So how tall would that be? 20 inches. Okay. Then what height plants would we expect in the F2 generation? So what we could do is we could do our Punnett square, right? Remember with our big A, big B, little b, gametes on the top and on the side, we'd get all of our different possibilities. And that works out to 1 that's all dominant, right? 2 that's big A, big B, little b. 2 that's big A, big B, etc. Okay? All right. So we already said that if you're homozygous dominant, you're going to be 30 inches. What if you have 3 dominant alleles and 1 recessive? How tall are you? 25. What if you have 2 dominant alleles? What if you have 1 dominant allele? And no dominant alleles? So how many different phenotypic classes are there? 5, right? So we have 2 loci, now we have 5 classes instead of 3. So if we added another loci, we'd have even more classes, and another loci, we'd have even more, right? So this here is just an example of this here. It's the same slide I just showed you. Now this is an example of that, right? If we have 2 loci, right, you could either be 10, 15, 20, 25, or 30 inches tall, right? So the more loci you add, the more different phenotypic classes can be involved. So if you look at this here, this many loci and phenotypic classes, what does that look like to you? It's a normal distribution. So we're going to be assuming a normal distribution in a lot of these cases, okay? Now, it's interesting to think about the idea that a single genotype may produce a range of phenotypes. So in this case, right, if you fall right here on the height scale, you don't know if you're a tall big A, or a short big A, little a. So sometimes it can be difficult to match the genotype with the phenotype because one genotype can lead to actually a range of phenotypes because of those environmental influences. Okay, we're going to assume that most quantitative traits follow a normal distribution. So hopefully this looks a little bit familiar. It has a little bit, tiny bit of statistics, hopefully over time. So this is the mean in the center, or the average, right? So a normal distribution is the bell-shaped curve with the average in the center. Okay? If we go out 1 standard deviation away from that average, that encompasses about 66% of our population. If we go out 2 standard deviations, that encompasses about 95% of our population. If we go out 3 standard deviations, that encompasses about 99% of our population. So the standard deviation affects the width of the curve. So if the standard deviation is very small, right, your curve is going to be very skinny. The standard deviation is very large, your curve is going to be a lot fatter. Right? So the mean, right, is just the average. The variance, you can calculate of a population. I'm not going to ever ask you to calculate a variance or a standard deviation. I would give that to you. The important thing to know, the variance is the square of the standard deviation. And the variance, this is what the term looks like. Standard deviation, in this case, is S. So if you looked at these 3 curves here, which curve has the greatest or largest variance? So yes, the blue curve, right, is the fattest curve. It's the widest curve. It has the largest variance. So that's really the thing I really want you to think about when we think about distributions. We are gonna use some of this information in doing some calculations later, but we're not gonna go into a lot of statistics with variances and standard deviations. What I do want you to realize, though, is that the larger the standard deviation, the wider the curve, right? And what that means is that if there's a very small standard deviation, there's not a lot of variation, there's not a lot of variance in your population, and there's a lot more individuals that are close to the mean, right, in red. If there's a big standard deviation or a big variance, there's a wide range of individuals in your population, right? So there's going to be less individuals that are close to the mean and more that are spread out amongst the population. So distributions can have the same mean, but very different variances. So this is an example of 3 different populations that all have the same average, but very different distributions. So knowing the variance can tell us the width of the curve. Now, quantitative traits, although they can give us a range of phenotypes, we can say that there's one that's a discrete phenotype. So an example of this might be something like blood pressure. Right? So there's a wide range of blood pressure. Blood pressure is determined some by genetic factors, some by environmental factors, right? But at some point, when you're above a certain threshold, we will say you have high blood pressure, right? So there's a range of phenotypes, but one phenotype may be classified as diseased or disordered, right? And anything above that would be classified as diseased. So just as a kind of hypothetical example, let's say there's 3 genes. Let's say there's no environmental effects on this particular phenotype. If you have a dominant allele at a locus, it gives you 5 units of risk. But if you have a recessive allele at that locus, you only have 2 units of risk. If you have 25 or more units of risk, you're classified as having the disorder. Okay, so let's think about this. What is the greatest number of risk factors you could possibly have? How many dominant alleles could an individual have if we're talking about 3 loci? 6, right? What is the lowest number of units of risk you could have? 12. Okay? If we had a cross of those 2 individuals listed there, Is it possible that any of their progeny would have the disorder?
What did you say the threshold was?
25. 25 or more. Okay, so if the question is, could anyone have the disorder, I would say let's try to figure out what is the most possible risk factors that they could pass on, right? So at the A locus, what's the riskiest genotype that their offspring could have? Big A, big A. What about at B? Big B, big B. And what about at C? Big C, little c. So each of the dominant alleles is 5, and the recessive is 2. So 5 plus 5 plus 5 plus 5 plus 5 is 5, 10, 15, 20, 25, 27. So 27 is greater than 25, so yes, their offspring could have the disorder. All right, questions so far? Okay, so if we're thinking about quantitative traits or traits that have a range of phenotypes, and we're trying to think about the idea that these phenotypes are due to both genetic factors and environmental factors, what sort of industries or occupations might be really interested in traits that are due to genetic or environmental factors and how we can increase or decrease those particular traits? Right, so if you are any sort of farmer, or if you're looking at livestock or anything of that nature, you may be interested in kind of adjusting your output and wondering if can we do that based on the genotypes, or do we need to affect the environment? Okay, so for the rest of the class today, we're really going to be thinking about your farmer friend and his cows. Okay, so he's noticed that some of his cows produce a lot more milk than other cows. And he's wondering, why is this? Why do some of these cows produce a lot of milk and other cows don't produce as much? Is it genetic? Are there genetic differences between your high producers and your low producers? If so, you could do some selective breeding, right? We could take those that have high production and breed them so that we have offspring that also have high production, right? Is it environmental? Right? Does it depend on the time of day they get fed, or when they are milked, or if they're closer to the windows, or I don't know, like other environmental things that might be beneficial to them? If that's it, then maybe you can like add more windows, or feed them at varying times during the day, or something that might be beneficial to making that change. So he wants to figure out, why are his cows different? Why do some cows produce more milk than other cows? Why is there this variation in his population? Okay, so he's like, you took genetics, you help me. Can you help me figure out what's going on with my cows, right? So you are thinking back and you remember we talked about these flowers, right? These flowers in their variation in their tube length. And we talked about how when we have a pure breeding strain, all of the variation is due to the environment. And when we have a heterozygous F1, all of the variation is due to the environment. And that in our F2 generation, when we have a range of genotypes, our variation is due to both the genotype and the environment. And you're thinking, his cows are probably this situation here, right? They're not all the same genotype. There's probably a range of genotypes in his population. So his variation is due to both the genetics and the environment. So how do we figure out how much is genetic and how much is environmental? Well, it turns out that this genetic variation is due to both additive effects and dominance effects. So, starting to put together some kind of formula-looking things here. So phenotypic variation is due to both the genotypic variation and the environmental variation. Or your phenotype for these quantitative traits is due to both your genotype and the environment. The genotype is due to both the additive effects and the dominance effects. So what are additive effects and dominant effects, right? Additive effects are the average effect of substituting big A for little a in a genotype. Dominant effects are due to particular combinations of alleles. So if the value of big A, little a is exactly in between big A, and little a, your dominance effects are 0. Okay, so what does that mean? So here's an example, hypothetically. Here's our genotypic variation, which is additive plus dominant effects. So if big B is 6 inches, big B is 4 inches, and little b is 2 inches, this is all additive effects because the heterozygous individual is exactly in between the two homozygotes, right? It depends on how many big B's we add, right, to get our phenotype. Kind of makes sense. We add 1 big B, we get 4. We add 2 big B's, we get 6. Dominance effects have to do with the presence of dominant alleles. So if the heterozygote is exactly the same as the homozygous dominant, then that's all dominance effects, right? It doesn't matter how many dominant alleles you have the same phenotype. And then oftentimes what we have is something a little bit in between, where being homozygous dominant, you're 6, and homozygous recessive, you're 2, but heterozygous, you're 5, which is like in between, but not exactly in between. That's a combination of additive and dominant effects. Now, why this is important to us, and one reason why this is important to us, is that the additive genetic effects primarily determine the resemblance between parents and offspring. So we're going to come back to that in a little bit, right? Think about if we're going to do a breeding program, we want a lot of additive genetic effects because we want the offspring to look just like their parents because we picked their parents to breed for a reason, right? And so that's why we chose them. We hope the offspring look just like them. Okay, so going back to that sources of phenotypic variation, same slide I showed you 2 slides ago. The phenotype is genotype plus environment. The genotype is due to both additive and dominant effects. And additive effects is the average effect of substituting big A for little a. Dominant effects has to do with the relationships with the dominant alleles. If the value of big A, little a is exactly between the two homozygotes, the additive effects is 1 and dominant effects are 0. Okay. We cannot look at a single individual and determine the values for G, E, A, and D. We have to look at a population, right? So if we look at a population and partition the variation into these different places, we can figure out what is the variation that we see, right? So in most populations, we're going to have some genetic effects, some environmental effects, some additive effects, and some dominant effects. So now we're going to make these look more like formulas, right? But again, when we're looking at these formulas, I want you to remember the concepts behind them. And if you remember the concepts or ideas behind them, it'll be really easy to remember the formula, right? So V sub P equals I'm sorry, I didn't listen to your question. Can you say it again?
Yeah.
Four. Okay. Hopefully that fixed it. So I apologize on this recording if there's a dead spot because the batteries just died. Okay, so the phenotypic variation or phenotypic variance is the genetic effects plus the environmental effects plus the G by E, but we're not actually going to include that. Okay? Also, epistatic interactions are important. So epistatic interactions are part of this phenotypic variation. So the phenotypic variation is the genotypic variation is the additive effects plus the dominant effects plus the epistatic effects. Okay, so remember, epistatic effects are when you have a particular genotype and it can mask a second genotype. So when you have a particular genotype at a particular location, it can mask a genotype at a second location. So again, it's really important when you're thinking about it and doing these sorts of problems, but we're not going to include it in our calculations because it overcomplicates things. Okay, so I do want you to know that G by E is a thing and epistatic effects are a thing, but we're not actually going to include them. Okay, so here you are. Now you're telling your farmer friend, okay, the phenotypic variation that you're seeing in your cows is due to both genetic variation and environmental variation. And the genetic variation, that is, can be subdivided further into additive effects and dominant effects. And so he just looked at you and said, well, but so what does that mean, right? Does that mean that should I breed my cows or should I add more windows? Like, what am I supposed to do here? I don't know. So you say, okay, what we need to figure out is what proportion of this phenotypic variation is due to genetic effects. Because if it's all due to the environment, you don't need to breed them special. You need to, feed them differently. Or if it's all due to genetics, then breeding them will be very beneficial, right? So how much of this variation is due to genetic variation? So how are we going to calculate that? How are we going to figure that out? Well, if we think back to our flower tubes, we could try to figure out what the phenotypic and environmental effects are in different populations and maybe use that to make some sort of calculation. Ultimately, what we want to calculate is what we call the heritability. The heritability is the proportion of the phenotypic variance that is due to genetic effects, which is basically what we just said. We want to know how much of this variation is due to genetics, right? So the broad sense heritability, or H squared, is V sub G over V sub P, or the genetic variance over the total phenotypic variance. The narrow sense heritability is the additive genetic effects over the total phenotypic variance. Now you may think, why do we need to know the narrow sense heritability? What benefit is that to us? Remember, I told you that the additive genetic effects are really what determines if the parents look like the offspring or if the offspring look like the parents. So really if you're trying to do some sort of breeding program, you really want to know the narrow sense heritability, right? What are the chances, right, that my offspring, or how likely is it that the offspring are going to look like the parents or have the same phenotype as the parents, right? If I pick a high producer, are their offspring going to also be high producers, right? That's what we're trying to figure out. Okay. So heritability ranges from 0 to 1. If the heritability is 0, there's no genetic effects at all influencing this phenotype. If the heritability is 1, there are no environmental effects influencing this phenotype. And you may hear heritability used in a lot of different contexts. So we can look and see heritability of certain things in human populations. So you can say things like autism has a certain heritability, or schizophrenia has a certain heritability, meaning the variation that we see in the population has a large proportion that's due to genetics, but also some proportion is due to the environment. Yes.
So you talk about progress at the higher heritability. What progress we can expect from selection breeding program? Like progress as in you get more of your desired effects. Like
More phenotypic change over generations. So a higher heritability, and we're going to do some examples with some numbers that'll probably make it make a little more sense. But if you have a higher heritability, if you start breeding together really tall, organisms, then you're going to have more tall offspring, and over time the whole population will get taller. If it's a low heritability, you can breed together tall individuals, but the offspring aren't going to be any more tall than the ones that were in the generation before. And again, we're looking at a population. So we're looking at the variation in a population. How much of that variation is due to genetics and the environment, not in an individual. So we're not going to say that 90% of your height is due to your genes and 10% of your height is due to the environment. We can see the variation that we see in height is 90% due to genetics, 10% due to environment. Okay, so you wanted to figure out what's the heritability of the milk production in these cows, right? Trying to bring it back to the cows. Okay, so you remember we talked about how if we have a pure breeding line, right, all of that variation is due to the environment. And then if we cross together two pure breeding lines, we still have a genetically uniform population, so all of that variation is due to the environment. And then in the F2, now if we cross these F1s together, now we have variation due to the genotype and the environment. So what if we did this experiment and we kept it in the same environment, right? So like we planted the bushes all in the same field, right? Or we, grew up the mice all in the same, room. If we kept our environment the same, could we use this to estimate the variation that's due to the genotype for this trait? So how would we do that? So if we assume that the environment is uniform, we can take the variation in phenotypes of parental strain 1, parental strain 2, and the F1, and use that to estimate the average environmental variance, right? So we're going to take those three variances and divide them by 3, take the average. That's going to give us our environmental variance. Now, assuming that stays the same for our F2, then we're going to say the variation of phenotypes in our F2, or the phenotypic variation, remember that's V sub G plus V sub E, right? V sub E we're going to say is the same as what's up here, so we can use that to estimate our V sub G. Now, do you remember what the formula for heritability is? Right, so heritability is the proportion of the variance that's due to the phenotype. So our heritability is the V sub G over V sub B. H squared is the term. Don't square anything. That's just the term is H squared. Okay, you guys ready to try one? Maybe. Okay, so here we have two pure breeding parental lines. The variance in parental line 1 is 3.3. The phenotypic variance in parental line 2 is 3.7. We cross those together to get a heterozygous F1, which has a variance of 5, or a phenotypic variance of 5. We take that heterozygous F1, cross it together, get a F2 that has a phenotypic variance of 20. So can you estimate the broad sense heritability for this trait? Okay. All right, so here's our problem. Okay, so remember, the phenotypic variance is the environmental variance plus the genetic variance. But in parental line 1, parental line 2, and the F1, what is the genotypic variance? 0. They all have the same genotypes as one another. In parental line 1, they're all pure breeding. They have the same genotype. So the parental line 1, parental line 2, and the F1, all of the variance is environmental. All of the variation we see is due to environmental factors. So we are going to estimate the environmental variances by averaging those three numbers. Okay? So we're going to take 3.3 plus 3.7 plus 5 and divide it by 3. So what did you get when we did that? 4. Okay, so in our F2, right, the V sub P of the F2 is 20, and this is the V sub E plus the V sub G. And we just calculated the V sub E is 4, so what's our V sub G? 16. So V sub G equals 16. Now our heritability, how do we calculate heritability? It's the proportion of the phenotypic variance that's due to genetic effects. So we would say 16 over 20. And so what does that give us? So we're using the variance in those three populations to estimate the variance in the F2, assuming the environment is pretty much the same or very similar. So our environmental variance is the average of those three. So our environmental variance is 4. Right? So in that F2, our genetic variance is that environmental variance. We're assuming the constant environment is 4, so our genetic variance is going to be 16. So our broad sense heritability is 0.8. Or in that population, 80% of the variation is due to genetic effects and 20% is due to environmental effects. Okay, so you're explaining this to your farmer friend, and you're saying if we have 2 pure breeding lines of cows, we can take the variation in phenotypes of those, and then we can cross them together to get the heterozygous F1 and take the variance in that, and then cross that together and get the F2, and then we'll have the variation in phenotypes there, and then we can use that to create the broad sense heritability. Think that's a good plan? Kind of long and complicated, right? Like he doesn't want to have 2 pure breeding lines of cows and cross them together to get heterozygous F1 and, no. There's got to be another way to do this, right? So we can do this in a slightly different manner. So we still want to calculate our broad sense heritability, but there's a different way to do it if we have a large population and we're trying to see what happens if we select for a particular phenotype. Okay, so what we have here, so look at the picture on the right first, okay? So here in the solid line is our initial population. There's a normal distribution. Some cows produce a lot of milk. Some cows don't produce very much milk. Here in the middle, X sub O, that is the average milk production of cows in that population, okay? So then what we're going to do is we're going to pick some of those cows and breed them. So he's going to pick some of the top producers. We're going to pick all these this population shaded in pink here. That's the group that we picked to breed. So we chose these cows to breed, and the average milk production of the cows that we chose is over here X sub S. Okay, so we bred those cows, then we have the next generation. So in the next generation, That population is here in the dotted line. Okay, so before we look at the numbers, what do you notice about the difference between the population in the solid line and the population in the dotted line? Right, it's shifted, right? So we've actually changed the phenotype in our offspring, right? And so the average milk production in the offspring is this X sub 1. Okay, so let's put some numbers to this. His cows originally, his average milk production is 80 liters per week. And I was informed by a student in my previous section that that's actually a really horrible milk production for cows, but I had no idea. So I'll change that for next semester. Evidently, that's not so good. But anyway, so he really needs your help, right? His cows aren't doing that great. So his original population is 80 liters per week. So he chooses some cows to breed. And again, the cows that he chose are represented by the shaded pink area under the curve. And the cows that he chose have an average milk production of 120 liters per week. Then on the F1 in the next generation, the offspring have an average milk production of 100 liters per week. So he's made a difference, right? He's increased the milk production in his cows. Okay? So can we use these numbers to calculate the heritability of milk production in his cows? Okay, I'm going to introduce a couple new terms to you here. One is the selection differential, which is S. And again, if you remember what it is, you'll remember how to calculate it. The selection differential is the difference between the average of those selected and the average of the population. Yes?
So out of all of these that we're going to be using, are any of them given to us? All of this?
There will be a couple I'll tell you about a little bit later, but none of the ones we've talked about so far. So the average of those individuals we've selected minus the average of the initial population. So in this example, what is our selection differential? What's the average of those we selected? 120. What's the average of the original population? 80. So our selection differential is 40. Pretty simple, right? Okay. The next thing we need to calculate is our response to selection. And again, if you think about what it means, it's going to be really easy to figure out how to calculate it. The response to selection is how did the population respond to the selection that you put them under. So we take the average of the next generation minus the average of the initial generation, right? What was the change from one generation to the next? So our X sub 1 is 100, X sub O is 80, so our response to selection is 20. So our heritability, we can calculate heritability by taking the response to selection over the selection differential. So our response to selection R divided by our selection differential S gives us our heritability. In this case, this is giving us our narrow sense heritability because remember what I told you about whether or not the offspring look like their parents is primarily due to additive genetic effects, and narrow sense heritability is involved in additive genetic effects. This will also be called, sometimes in the book and other places, it's called realized heritability because it's calculated based on a real situation. So narrow sense or realized heritability, we use those interchangeably. So in this case, in his population of cows, what's the narrow sense heritability? 0.5, right? 20 out of 40, 0.5. So the heritability for milk in your farmer friend's cows is 0.5. All right, you ready to try one yourself? So your friend found out that you're great with cows, so they wanted you to help them with their chickens, right? So he has some chickens. His average egg production of his chickens is 229 eggs per year. So he selected some individuals to be parents, and he selected individuals that were 293 eggs per year. And then in the next generation, the offspring produced 253 eggs per year, So what is the heritability, the realized heritability, or the narrow sense heritability in this population? So that's what the question is asking. I'll leave it off the screen so you can see the text. But okay. So let's go ahead and start walking through this. So the original average egg production is what? 229. The average production of those he selected to breed are 293. And then the average egg production in the next generation? 253. Okay. Right, so S, our selection differential, is going to be our 293 minus 229. And so what'd you get for that? 64. Okay, and then our response to selection is our average in the next generation minus the original, so that's 253 minus 229. So what did we get for that? 24. And then our heritability is going to be R over S. So in this case, our heritability is 0.375. So original average 229, average of those selected 293. Average in the next generation 253, right? Our S is 64, R is 24, and our heritability is 0.375. Okay? Questions about that one? Okay, so let's say that your farmer friend is like, okay, well this is great. We figured out that the heritability in my cows is 0.5. So what if I continue with my breeding program? What if I do it another generation and another generation? Can we make predictions as to what's going to happen over time if we know that heritability? What if we know the heritability and he's like, I just want to take like the top 10% of my cows and just breed the top 10% and figure out is that going to be beneficial over time, right? We can definitely do that. And to do that, we're going to have to use a couple of new terms. Okay, so here is the same population distributions I've showed you before, right? In the solid line, that's our original population. the mean of the original population is X sub O, or for whatever reason, for the purposes of these formulas, they give them as mu. Then our F1 is in the dotted line, right? And the mean of the offspring population we have is X sub 1. Under here, the proportion that's selected is shaded in pink. But in this case, we're selecting a percentage. Okay? So if we're selecting a percentage, we may not know the average value or the average phenotype of those that we've selected. So what we may need to calculate is a truncation point. A truncation point is the point at which everything past that is the proportion that is going to be selected. Okay? Now we're still going to need to calculate what's going to happen in the next generation. So in this case, Farmer Fred wants to know, if I keep doing my breeding, what would happen in the next generation? So in order to figure out your X sub 1, what do you need to know? What value do we need? We need R, right? Because R is the difference between X sub O and X sub 1, right? So we're going to try to figure out what is R. Well, based on what we learned before, heritability is what? Narrow sense heritability. How do we calculate that? R divided by S. So if we rearrange that formula to figure out R, what is R? Heritability times S. Okay? So the initial population mean is X sub O or mu. We're going to use the standard deviation in this particular calculation. If we calculate the truncation point, that's labeled X sub T, and we are going to give you that formula for the truncation point. The selection differential, because we don't know the average value of those selected, we're going to have to calculate that. That's going to be I times the standard deviation. We will provide you with that formula as well. I and Z will be chosen from a table of standardized values. The response to selection, we already know, is going to be heritability times S. And the mean of the next generation is going to be our original mean plus the response to selection. All right, so let me show you how we're going to use this. Right? How do we use this to actually figure anything out? So what we want to do is we're going to use Z and I from the table to be able to find out our selection differential and our truncation point. Then to be able to finish answering the problem, we'll need to use our realized heritability. Now one thing that's important to note is that if we are going to make something smaller, so let's say we want to have a smaller percentage of fat, or we want to have a smaller, mouse or something. If we're selecting the left end of our curve, then our values of I and Z will be negative. Okay. All right, so let's help our farmer friend here and see if we can help him figure out what would happen if he continued breeding his cows. So if he selected the top 5% of milk producers, then what would happen in the next generation? That's what he wants to know. If he takes his top 5%, what will be the average milk production in the next generation? Okay, so one of the first things we want to calculate is our truncation point. So we want to know, who are the top 5%? Who are the top 5% milk producers in his population? Well, what's the standard deviation of milk production in this population? 18 liters per week. So the truncation point is going to be the mean plus Z times the standard deviation. The mean is 100. Z comes from the table. How do we know which column of the table to use? The proportion selected. He's selecting 5%. So we're going to use this column of the table here. So our Z is going to be 1.645. So Z times the standard deviation is 100 plus 1.645 times 18. So our truncation point is 129.61. So he's going to take every cow that produces 129.6 liters per week or more and use them to breed. Okay? The next thing we want to calculate, remember our goal, what we want to get to is we want to calculate R. Remember? And heritability is R over S. Do we know heritability? Yeah, we do. We already calculated that from the last situation. So we do know the heritability. We're assuming the environment's the same, so we know the heritability is 0.5. We need to figure out the R, so we need to find the S. S, the selection differential, is going to be I times the standard deviation. And again, I is going to come from this table. In this case, I is 2.063. Standard deviation is 18. So the selection differential is 37.174. Now again, what he really wants to know is the response to selection. Like, what's the difference? What's going to happen in the next generation? So to figure out our response to selection, right, we're going to take the heritability times the selection differential. Heritability is 0.5. Selection differential, we just calculated 37.174. Our response to selection is 18.567. So if your farmer friend selects the top 5% of milk producers, what will be the average milk production of that next generation? $118.567. The response to selection is saying we're going to move the average 18.5 liters per week. So it was 100. If we do this breeding process, we're going to increase it by 18. Questions? Are you following enough to have a question? So I will definitely recommend, I'm going to go back, I'll come back here for a second, is look at this picture here, right? And if you really understand what this picture is showing us, right? The original population with the original mean, right? The proportion selected, we could add on here x sub s, which would be the average of those selected, right? The average of those selected minus the initial average is s. We can also figure out that s by using the formula we were provided if we're selecting a percentage, right? The response to selection, that's really important, is the difference between the original and the next generation. So sometimes we're going to be, we'll know what the average of that next generation is, and we're going to be calculating the heritability. Sometimes we're going to know the heritability, and we're going to want to predict what the response is. We'll say, look, if the heritability for this trait is 0.8, what's going to happen if I do this breeding? Or if the heritability for this trait is like 0.2, what's going to happen if I do this breeding? Is it worth it? Is it worth my time? Or should I do something else, right, to make a difference in my population? So, you're helping your farmer friend. He's increasing this milk production in his cows. Can he do that forever? Like ad infinitum? No, there's a limit, right? You can't just, at some point, the cows cannot produce any more milk, right? It won't be healthy for them to produce more milk than what they're producing. So over time, right, the selection response may be limited. You may reach kind of a threshold where you can't go past it. This could be, for one, due to losing genetic variation, right? As we are continuing to breed and breed and breed to pick for these particular traits, we may end up getting a very homozygous population, which may not be beneficial. The extreme types may not be healthy, right? Being super, super high milk producer may actually not be a very healthy cow. So one example is in mice, there was an experiment done to select for smaller and smaller and smaller mice, and it got to a point where the mice were sterile. They were so small they could not produce gametes that were viable. Right? So at that point, you're kind of stuck because you can't make any more. Two desirable traits may be negatively correlated. So for turkeys, when you select for rapid growth and increased body size, right, so farmers want turkeys that grow fast and fat, that actually is correlated with decreased fertility, decreased egg production, as well as decreased egg hatchability, which kind of makes sense. If the turkey's putting all its energy in getting big and fat fast, it doesn't have a lot of energy to put into making eggs that survive. But that can be a problem, right, because you need more turkeys, right? You just don't want the one turkey. You want more turkeys after that one turkey, right? So we do need eggs to be produced. So most commercial turkeys, they have to artificially inseminate and use other means of getting the offspring because of the correlation, the negative correlation between body size and fertility. So an example in corn where they took basically a strain of corn and they selected for both high and low oil content. So maybe we want corn that is very high in oils that can be used for various processes and things, right? Or maybe we don't. Maybe you don't want corn that's high in oil. Or maybe if we have two lines of corn, one that has high oil content and one that has low oil content, maybe we can use that to figure out the genetic differences, right? How could we do that? What if we sequenced the DNA of these corn that have high production and we sequenced the DNA of the corn that has low production and compared them and tried to figure out what are the alleles that are different, right? And they found 20 different genes that were involved in this process, that were different between these two strains involved in oil production, right? So over 20 different genes are involved in that particular quantitative trait. Another example, here's one in Drosophila where they selected for bristle number, and you can see after about 20 generations, it leveled out because you can only have so many bristles on the back of a fly. The other thing is that a change in value for one trait may be accompanied by the change in value for another trait if the genes affect more than one trait. So oftentimes we have genes that affect more than one thing, right? It's called pleiotropy. So the correlation can be positive or negative. So here are some examples from a table in your book, right? So if you look at a pig, the more weight they gain, the more fat they have, back fat they have. If you look at a chicken, the bigger they are, the bigger their eggs are, which might seem really good, except the bigger they are, the less eggs they produce, right? Which makes sense. If you're making really big eggs, you're not going to make as much, right? So when you're trying to, increase certain traits in a particular line or organism, you kind of have to think about what are the other traits that you're going to be affecting as well, right? So for a mouse, if you increase the body size, it also increases the tail length, which kind of makes sense, but it's just something to consider as you're looking at these different traits and changes that we're making in them. So we'll stop here with quantitative genetics, and after the break, we're going to talk about mapping these genes and how do we find them, and how do we do this stuff in humans. So have a great Thanksgiving holiday, and I will see you when you return.